This video goes along with chapter 6 in your book. We will be looking at cell structure and function, which covers a lot of different things. You can read through these topic statements if you wish, but that's on you. So starting off, how do we even know what cells look like? That is through microscopy. So the first microscopes were around in about 1590 and they kept being refined. The cell walls were first seen by Robert Hooke in 1665 because he was looking at dead bark from an oak tree. So all he could see was the outline of the cell wall, and that was it. Leeuwenhoek made a better microscope that was able to see living cells, specifically pond water. And then we had the development of light microscopes, which uses light passed through the lens to be able to see the specimens. These are the kinds you are familiar with and have used before. And then the fancy microscopes called electron microscopes were, initially around in the 1950s and have been improved several times since. There are two types: scanning electron, which gives you a detailed topography of a specimen, and transmission electron, which lets you see the internal structure of cells. To the right, you can see the size range of cells. Most of them, as you can see, are quite small, and then there's even some where you can see like ribosomes are, maybe 20 nanometers. And then all the way down to the atom. So depending on the cell type, the cell size will be different, and we will look more at the implications of cell size another time. Cells are the basic structural and functional unit of living things, and there are two broad types: prokaryotic and eukaryotic. Recall that there are three domains of life. Prokaryotic cells include the domains bacteria and archaea, and the eukaryotic cells are from the domain eukarya, which is where protists, fungi, animals, and plants reside. Let's start by comparing prokaryotic and eukaryotic cells. All cells have a plasma membrane, aka cell membrane or phospholipid bilayer. They all have cytosol, which is a semi-fluid, jelly-like substance where the components are suspended. They have chromosomes. In eukaryotes, they are linear, and they are circular in prokaryotes. All cells have ribosomes to make proteins, and cytoplasm, which is the interior of the cell. So you can see to the right here the generic structure of a prokaryotic cell. It has flagella for movement. It has a cell wall, a plasma membrane, some ribosomes, and a nucleoid, which is just the area where the DNA is in, even though it is not enclosed in a nucleus. Some differences are the location of DNA. In eukaryotic cells, DNA is found in a nucleus, but in prokaryotes, it's concentrated in a region that's called the nucleoid. Eukaryotic cells have membrane-bound organelles that are suspended in the cytosol. Prokaryotic cells do not have them, but they will have regions where reactions take place. So there will be a region where respiration takes place, or perhaps form of photosynthesis. It just depends. They are relatively different size-wise. Eukaryotes are generally much larger, but that is not always the case, depending on the cell type that it is. Cell size relates to function because cell metabolism sets the limit on cell size. So for each square micrometer of plasma membrane, a limited amount of substance can cross per second, which means the surface area to volume ratio is critical. So surface area grows proportionately less than its volume. Smaller objects have a greater ratio of surface area to volume, which explains the size and shape of cells. They are microscopic, and like with nerve cells, they are narrow and elongated. But we will do some practice with this in class. Eukaryotic cells have internal membranes that divide the cell into compartments or organelles. Each compartment has a specific metabolic function. And then there are, of course, proteins that attach to the plasma membrane. Here you can see a diagram of an animal cell and a plant cell. We will go through the differences and components of what is in these cells next. Starting with the nucleus. The nucleus contains most of the genes in a eukaryotic cell, and it is surrounded by a nuclear envelope. The envelope does have pores to allow things like mRNA to go in and out. It does have a double membrane, so it has a lipid bilayer with some proteins in it. In eukaryotic cells, DNA is organized into chromosomes. Each chromosome contains a long DNA molecule with many proteins so that it can be compacted enough to fit inside the nucleus. There are proteins that help it coil and condense. And when we talk about chromatin, that is DNA plus its associated proteins. The nucleolus is a region of the nucleus where rRNA is synthesized. rRNA are the subunits, there are two, that make up each ribosome. Each ribosome will have a small and large subunit that's assembled after it leaves the nuclear pore, and then it can start to make proteins. One cell process that occurs in the nucleus is the synthesis of mRNA. So DNA holds the instructions for mRNA synthesis. Those mRNA molecules leave the nucleopore, find a ribosome, and then is translated into a polypeptide. We will go a lot more into the details of that process much later. Ribosomes, like I said, are made up of a large and small subunit, and the size of the subunits between prokaryotes and eukaryotes are different. They carry out the process of protein synthesis. They are not membrane bound, so they are not considered an organelle, which is why we also find them in prokaryotic cells. Cells that need to make lots of proteins will have lots of ribosomes. They will either be free or attached. Free ribosomes are suspended in the cytosol, and attached or bound ribosomes are attached to the endoplasmic reticulum, which makes it the rough endoplasmic reticulum. And these ribosomes can play either role, so they can be free-floating and then attached, be attached and then free-floating. It just depends on the situation. Ribosomes that are bound to the endoplasmic reticulum are destined for insertion into membranes and are packaged, as we will see later. The endomembrane system is next, which includes the nuclear envelope, endoplasmic reticulums, Golgi apparatus, lysosomes, vesicles, vacuoles, and the plasma membrane. The roles of the endomembrane system are to synthesize proteins, transport proteins into membranes or out of the cell, metabolism and movement of lipids, and detoxification. They either have direct physical continuity or vesicles to transfer the membrane segments, and they will all vary in structure and function. Starting with the endoplasmic reticulums, which are an extensive network of membranes, as you can see from the picture on the right. They are a network of membranous tubules and sacs that are continuous with the nuclear envelope. And there are two distinct but connected regions. One is called the smooth endoplasmic reticulum because it lacks ribosomes. Its function is for lipid synthesis, metabolism of carbohydrates, detoxification, and storage of calcium ions. This does include synthesis of vertebrate sex hormones like estrogen and testosterone. The rough endoplasmic reticulum has bound ribosomes on it, and the cells secrete proteins produced by the rough ER, and they end up wrapped in vesicles. Next is the Golgi apparatus, which you can see a lovely diagram of here to the right, receives transport vesicles from the endoplasmic reticulum. So these ER products are modified, stores, and sent to other locations. It consists of a group of associated flattened membranous sacs. Vesicles from the ER add its membrane and contents by fusing with the Golgi membrane. Pectins and non-cellulose polysaccharides will be produced here as well. And things leaving the Golgi will depart in a vesicle and eventually fuse with the plasma membrane. So those vesicles bind to the membrane of the Golgi, do what it's supposed to do, and then they are, vesicled out to go attach to the plasma membrane. Lysosomes are a membranous sac of hydrolytic enzymes that digest macromolecules. Lysosomal enzymes work best in the acidic environment inside the lysosome. So even if the pH overall of the cell is, close to normal, the pH inside the lysosome will be a little more acidic. The enzymes that can be released either from a break or a leak in the lysosome will still work in the cytosol, but they will not be as effective. Hydrolytic enzymes and the lysosomal membrane are made by rough ER and transferred to the Golgi for processing. And they carry out the intracellular digestion. Then digestion products are released to the cytosol and used so we can recycle the cell's organic material. One disease associated with the lysosome is Tay-Sachs, where certain products are not digested properly. And lysosomes are not found in plant cells. This job is done by the vacuole in the plant, but animal cells have tiny vacuoles and lysosomes to carry out those functions. Vacuoles are large vesicles derived from the endoplasmic reticulum and Golgi. And they are selective in transporting solutes, and they have a variety of functions in different cells. Amoebas, for example, have a food vacuole from phagocytosis, where they essentially eat things Pac-Man style to form a food vacuole. A contractile vacuole can pump excess water out of cells in unicellular freshwater organisms. In plants and fungi, they are for hydrolysis. In plants, they also can store organic compounds that are poisonous to animals, and pigments to attract pollinators. A plant has a central vacuole that is the main repository of inorganic ions, helps with cell growth, and absorbs water. In plant cells, it is the largest structure, as you can see from the picture, but animals will generally just have a few small vacuoles instead of one large one that takes up, roughly a third of the cellular space. Next, we will talk about mitochondria and chloroplasts. Mitochondria is not the powerhouse of the cell. It is, however, the site of cellular respiration. Chloroplasts are found in plants and algae and are the site of photosynthesis. Both organelles have similar evolutionary origins for how they came to be in eukaryotic cells. And this is called a theory of endosymbiosis, or the endosymbiont theory. So mitochondria and chloroplasts have similarities with bacteria. And this theory tells us that mitochondria and chloroplasts originated as prokaryotic cells, were engulfed by a host cell. The engulfed cell and its host then evolved into a single organism. So we know that the mitochondria came first because plants and animals have that, and then the chloroplast later. And this theory is consistent with those specific organelle structural features because they both have two membranes. They contain their own ribosomes. They have their own circular DNA, and they can independently grow and reproduce in a cell without getting a signal from the nucleus that it's time to do that. So you can see how that process occurred in the picture here, which I think is super awesome. Mitochondria are found in nearly all eukaryotic cells. Both of the membranes around the mitochondria have a phospholipid bilayer with its own embedded proteins, but it has a smooth outer membrane. The inner membrane has convoluted infoldings to form an intermembrane space in the mitochondrial matrix, which is, we'll talk about, the details and why that's important when we get to cell respiration in several weeks. But as a preview, the electron transport chain and ATP synthesis, which are big parts of cell respiration, do occur on the inner mitochondrial membrane, which you can see in the lovely picture here. Chloroplasts contain the green pigment chlorophyll. It is found in the green organs of plants and algae. It also has two membranes. Its membranous system of flattened interconnected sacs are called thylakoids. The thylakoids are stacked, and each stack is called a granum. The plural form of that is grana. Contains chloroplast DNA and ribosomes, and this is where the light reaction of photosynthesis occurs. It is a specialized member of a family of closely related plant organelles called plastids, which include chloroplasts, chromoplasts, and amyloplasts. It also has fluid with the inner membrane called stroma, where the Calvin cycle, which is the second part of photosynthesis occurs. And here we can see a lovely picture of a chloroplast. Next, we will talk about peroxisomes, which is an organelle that contains enzymes that transfer hydrogen atoms to oxygen to produce hydrogen peroxide. It has a single membrane. It is for detoxification of alcohol in the liver. It can break down fatty acids into smaller molecules, but H2O2 is toxic to cells, so peroxisomes also contain an enzyme that converts hydrogen peroxide to water, and we don't know how they are related to other organelles. The cytoskeleton is the network of fibers that extend throughout the cytoplasm that serve a variety of mechanical, transport, and signaling functions. We're not going to go into a lot of detail about this here, but you do need to know the basic functions. Your book goes into way too much detail on this. Bacteria also have a cytoskeleton, but we're only going to focus on the eukaryotic cells here. They're composed of microtubules, microfilaments, and intermediate filaments. It gives mechanical support to the cell to help maintain shape, which is especially important for cells that don't have a cell wall. And some movements around the cell do involve the cytoskeleton. Microtubules are hollow rods constructed from tubulins. They are also found in cilia and flagella, which are the exterior cellular structures used for movements. They serve as tracks along which organelles with motor proteins can move, and they help separate chromosomes during cell division. Microfilaments are thin solid rods built from actin, and they can cause cell contractions. Intermediate filaments are only found in some animal cells, and they are specialized for bearing tension. So you can see some examples and more information about the cytoskeleton filaments here. And that is it.